My name is Aya. I'm a PhD student from Aarhus University in Denmark, and I would like to take you on a journey following the footprints of hybridization in North Atlantic eels. Hybridization creates novel genotype combinations that are subject to natural selection, and depending on their fitness effects, certain alleles may introgress more easily than others. Incompatibility between allelic variants from different species may reduce fitness or even cause inviability of hybrids. Environmental selection can also act against introgression. But introgression can also drive adaptive processes. Adaptively important variation may introgress even against the backdrop of genome-wide purifying selection. As a result, species that experience ongoing hybridization are expected to have porous genomes, with some regions that are resistant to gene flow and some regions that are more permeable. Angled eels represent remarkable cases of hybridization and incomplete reproductive isolation between species pairs. So the European eel and the American eel that inhabit the Atlantic Ocean are morphologically almost indistinguishable from each other, but they do differ in several life history traits, such as the length of the larval phase and their migration distance. But really the most fascinating thing about these two species might be that they are both genetically panmictic, meaning that each species consists of only one single population with completely random mating. Due to their life cycle, these species have no possibility for local adaptation, and therefore there is no genetic differentiation in these respective populations, despite them occupying very large geographic ranges. The species, they spawn in partial overlap in connection with the frontal zones of the Sargasso Sea. From here, ocean currents transport the larvae to their respective habitats on the continental shelves of Northern America and continental Europe. The larvae then transition through various life stages, either in freshwater or in coastal waters, and then several years or even decades later, the adult eels undertake the migration back to the Sargasso Sea where they spawn and then perish. Interestingly, first-generation hybrids between these two species have never been found anywhere outside of Iceland, not even in the spawning area. And only a few back crosses have been found in continental Europe and North America. So the specifics of hybridization in the Sargasso Sea are unknown, and no distinct hybrid zone has ever been found despite numerous sampling initiatives. The extent of hybridization and its effects are also still under discussion. The occurrence of a few individuals that appear to be this curious result of a first-generation hybrid backcrossing first to American eel and then their offspring backcrossing to European eel has raised an hypothesis of an actual hybrid swarm occurring in Iceland. The skewed distribution of hybrids has also given rise to a theory of a north-to-south gradient of introgression occurring in continental Europe, with an expectation of higher introgression levels in the northern European eels. I explored the patterns and the dynamics of introgressive hybridization and selection in North Atlantic eels through analysis of whole genome sequences from 49 European eels, 7 American eels, and 22 hybrids, of which 21 were caught in Iceland. To begin with, we assessed the hybrid status of these Icelandic eels to clarify if they really represent a swarm of complex hybrids and backcrosses. Next up, we wanted to resolve these issues regarding introgression outside of Iceland. So first, this involved testing if the genomes of these eels are porous, and we did that by identifying introgressed segments of American eel genome in European eels. Then we tested the hypothesis of a gradient of admixture occurring in continental Europe. And lastly, we then investigated if adaptive integration from American eel into European eel is ongoing by searching for chromosomal segments with signatures of both integration and positive selection. So first off, I utilized the software PC Admix for local ancestry assignment to produce what we call chromosome paintings. Essentially, this software divides the genome into small blocks, and then using PCA in combination with a hidden Markov model, it assigns the most likely ancestry to each block. And the end result is visualized here for chromosome 1. The faint white lines divide individuals from each other, and each individual is represented by 2 lines, one for each chromosome copy. Blocks or chromosome copies can then be colored either orange for European ancestry or blue for American ancestry. The topmost individual here is a first-generation hybrid. We can see that because it has 1 chromosome copy of either color, and there's no observable recombination events having occurred between these chromosome copies of different ancestry. In the same figure, there are also a lot of individuals that have 1 orange-colored chromosome copy and then 1 chromosome copy that is partially blue and partially orange, meaning that it is some type of backcross to European eel. And the purpose of creating these chromosome paintings to begin with was to explore how integration rates can vary across the genome, because we believe that this could provide some insights into how species boundaries are maintained in the face of ongoing gene flow. But at this point, it became apparent that the individuals we have in this study are mainly first-generation hybrids and first-generation backcrosses to European eel. And we were able to estimate that based on the genome length, the number of observable recombination events having occurred between chromosome copies of different ancestry, what we call ancestry switches, and then genome-wide ancestry proportions for each individual. And that is what this figure on the right shows. So the singular dot at the bottom is representing a total of 10 first-generation hybrids. They are equal parts of both species, and they have no observable recombination events or ancestry switches. The expectation for a first-generation backcross to European eel is that its genome is 75% European, 25% American, and with a genome length of about 1700 centimorgan, we expect to be able to see about 13 ancestry switches. And that's right around the point where the majority of the remaining individuals cluster. The inference of admixture timing was also corroborated by analysis of species diagnostic markers with new hybrids. So this means we weren't able to shed light on the genomic architecture of hybridization due to a lack of later generation hybrids. But the results here still provide some insight into the extent of hybridization in North Atlantic eels. It suggests a much simpler scenario than previous studies that have reported these complex backcrosses and even F2 hybrids, which are the result of first-generation hybrids mating. Here, most post-F1 hybrids are simple backcrosses in the direction of European eel, and while there are a few cases that may be more complex than that, this can hardly be considered a hybrid swarm. We also produced these chromosome paintings for the 49 European eels, and this showed that there were introgressed American elements in every single one, which suggests that yes, the genome is porous. On the contrary, we found no evidence for an introgression incline or a gradient. This plot here on the bottom right shows the 7 European sampling locations sorted by latitude, and there was no significant difference in the average individual American genome proportion between these locations. Generally, introgression seemed random, but there was this one instance where introgression occurred at a high frequency in a roughly 200 kilobase per section of chromosome 15. And this finding actually corresponds with a previous report of a possible inversion in this position. And I don't have time to go into much detail, but selection scans shown here on the left confirmed that this position is under positive selection in both species. Inspection of the decay of extended haplotype homozygosity plotted on the right showed that the American eel with individuals in red text are fixated for one genetic variant, the minor allele here shown in blue, and that introgression into European eel has occurred on multiple occasions. Moving on to the regions of the genome that appear less permeable, we and several before us have plotted FST along the chromosomes and found that there are these large islands of relative divergence. These regions have been regarded as barriers to gene flow. But when we looked into these regions on our chromosome paintings, we didn't observe any resistance to introgression. The proportion of introgressed chromosomal blocks in regions didn't differ significantly from the genome-wide proportion of introgressed blocks. Also, we observed that absolute divergence measured as DXY was not higher in these areas, but actually lower. And this feeds into a much larger discussion relating to the interpretation of high relative divergence measures. But it does suggest that these islands are not the result of reduced gene flow, but actually the result of reduced diversity. And this corresponds with the fact that the selection scans here on the bottom right show strong signatures of selection in the exact same regions. Because selection reduces diversity, and that means it affects the relative measures of divergence, but not the absolute measures. To quickly sum this up, our genomics approach provided insights into the dynamics of hybridization and introgression in North Atlantic eels. And our results showed that hybridization is a matter of simple backcrossing to parental species, not a hybrid swarm. It showed that genomes of Atlantic eels are porous, and introgressed segments of American eel genetic material is commonly observed in European eel. It showed that despite the skewed distribution of hybrids, introgression levels are similar across continental Europe. And lastly, identification of introgressed material that is simultaneously under positive selection in both species provided evidence for ongoing adaptive processes or adaptive introgression. And with that, I would like to thank the funding bodies of this project, my supervisor, all of my co-authors, and other collaborators.